And I think that if you want to be a good competitor, you need to be rewarded to be enoughly fit. Is it true? So it's my pleasure to introduce the next speaker, Manuel Guzman, coming from CIBERNED. He's a Spanish biologist at present, full professor of biochemistry and molecular biology at Madrid Complutense University. His PhD and postdoctoral research interests focused mainly on the study of the regulation of liver and brain lipid mechanisms and metabolism. And for the last 2 decades, he has been involved and interested in the study of active components of cannabis, the cannabinoids, to understand its molecular mechanisms and the way they may control cell generation and death. So please welcome with me Manuel Guzman for his lecture on neuroprotection by the CB1 cannabinoid receptor in the corticosteroid circuit. Thank you. The floor is yours.
So good afternoon, and thanks very much indeed to the organizers for this very kind invitation and for making this very nice event. Really, it's a good achievement. Well, in this talk, I will try to summarize what we have learned the last few years on one of the lines of research we are dealing with in our lab that implies the potential neuroprotective role of cannabinoid receptors, and I will give you as an example what happens in the corticostriatal loop. The talk doesn't intend to be comprehensive. It's a very brief flavor of what we are doing, and of course, I will be happy to take questions about technical approaches, etc. So as you most likely know, cannabis sativa produces a family of active ingredients, the so-called cannabinoids, that act in our body by mimicking the action of endogenous substances, of endogenous extracellular messengers that are therefore termed endogenous cannabinoids or endocannabinoids. These compounds are a family of arachidonic acid bioactive derivatives, and specifically there are 2 endocannabinoids described so far in the mammalian brain. One is the ester of arachidonic acid and glycerol in the SN2 position of glycerol. There is 2-arachidonyl glycerol, 2-AG. The amide of arachidonic acid and ethanolamine. Ethanolamide, arachidonyl ethanolamide, or more commonly termed as anandamide. Endocannabinoids act in our body, and of course, cannabinoids do so by molecular mimicry, by activating specific receptors, specific metabotropic inhibitory GIO couple GPCRs that are widely expressed in our brain. There are 2 types of cannabinoid receptors that have been cloned and characterized so far in mammalian tissues. One is the ubiquitous receptor that is extremely abundant, and we'll see later on in our brain, cannabinoid type 1 receptor, CB1 receptor, that is very highly expressed by many neurons and neuroglial cells, and also by peripheral tissues. Whereas there is another type of cannabinoid receptor that is more restricted, this cannabinoid type 2 receptor, to immune system cells and tissues, and in the brain it is mainly expressed in microglial cells. Cannabinoid CB1 receptor is one of the most abundant presynaptic metabotropic receptors in our brain. It's especially expressed by GABAergic terminals, for instance, GABAergic interneurons or medium spiny neurons in the striatum albucylatron, but it's also expressed at smaller amounts in other type of terminals, for instance, in glutamatergic terminals and other types of terminals. And this is, of course, the neurobiological basis for endocannabinoids to modulate a very large and wide array of processes in our body. Of course, those are the processes that are affected by cannabinoids derived from cannabis. And one of these processes, cell generation and survival, has been the main focus of our lab for the last 15 years. Well, in the specific context of CIBERNED, I would like to highlight that cannabinoids have been shown to exert neuroprotective actions in different animal models of brain damage and neurodegeneration. These are exemplified by this, and there are other types of models. And I would like to highlight as well that these are so far preclinical studies, as cannabinoid-induced neuroprotection hasn't been yet proven in the human brain. But I would also like to mention that there is clinical proof for efficacy of cannabinoids in the management of different symptoms of neurodegenerative disease. And as you most likely will have heard or know, cannabinoids have been shown to be capable of attenuating spasticity in MS patients, and cannabinoids have also been shown to inhibit convulsions in some refractory epilepsies in kids, pediatric epilepsies. These studies that have been conducted very widely in the last 10, 15 years by many different groups all over the world, including ours, have, however, been conducted by using systemic approaches. That is, for instance, giving cannabinoid drugs to animals or using, for instance, CB1 receptor knockout mice. But as I already mentioned before, cannabinoids can act via cannabinoid receptors located in different neural populations, including neurons with different opposing phenotypes, such as GABAergic or glutamatergic neurons, and also by different types of glial cells. So the goal of our group during the last 5 years, 6 years, have been to determine which are the precise pools of CB1 receptors that are involved in neuroprotection. Is CB1 receptor located in neurons and or in glial cells, in which type of glial cells, in which type of neurons, etc. And for this purpose, in the last year, we have developed a series of genetic and chemogenetic approaches to tackle this issue. And as I told you, I will give you an example of a circuit in which cannabinoids are very active in our brain. A CB1 receptor is very highly abundant in this corticosterial loop and controls the activity of this circuit that, of course, controls, in turn, very different and important neurobiological processes such as motor behavior, cognition, reward, affection, etc. In this loop, in this circuit, cannabinoid receptors are expressed by GABAergic neurons, especially by direct pathway and indirect pathway medium spiny neurons. Cannabinoid receptors are also expressed by glutamatergic projections that are synaptic onto both direct pathway and indirect pathway MSNs. Cannabinoid receptors are also expressed by astrocytes, both from the cortex and the striatum, etc. So we have many different pools of CB1 receptors that we are interested in analyzing which of them can be important and which cannot be important for neuroprotection. And in one of the most paradigmatic diseases of this circuit, as Sarah explained before, CB1 receptor, Huntington's disease, HD, CB1 receptor decreases at early stages of the disease, both in animal models and in patients. So, some years ago, we started to tackle this issue. What is the role of cannabinoid receptors in HD? And specifically, what is the role of cannabinoid CB1 receptor in MSNs, that are the cells that degenerate primarily, as you know, in HD? So to start with, we did an overall systemic approach, as I mentioned before, that people did, that was the state of the art some 6, 7 years ago in the field. So we crossed mice that are transgenic for a pathogenic form of human Huntingtin, exon 1, XS2 mice, so that's just a model for HD, for Huntington's disease, and we crossed them with CB1 knockout mice. So we generated HD-like mice without CB1 receptors. And to make a long story short, we found that upon deletion of CB1 receptors, these animals, these HD animals, these RCX2 animals, did worse than the lithomates that express 2 copies of the CB1 receptor. You can see here in the rotor rod that is worsened in the RCX2 animals compared to the wild types, but when we remove CB1 receptors from the RCX2 animals, they do worse compared to the CB1++ lithomates. For instance, another example, striatal atrophy is one of the hallmarks of Huntington's disease. It's also of Huntington's disease models, and you can see here that the atrophy that is observed in HD mice is worsened upon the loss of CB1 receptors. So that implied that this and many other experiments implied that CB1 receptors were protective in RCX2 mice, or in other words, that upon deletion of the receptors, the phenotype of the mice worsened. However, this is a global approach. What happens in MSNs, that are the cells that degenerate in Huntington's disease and are cells that express a very large amount of CB1 receptors? Of course, we expected that it was going to be those receptors that were important for neuroprotection, but you will see that that was not the case. What happens if we delete CB1 receptors in MSNs, specifically in MSNs, not in the whole body, not in the whole batch of cells in the body, in these R62 mice? So we generated R62 mice with the CB1 receptor gene flux, flanked by flux sequences. We injected an adenovirus vector that is encoding the recombinase under the control of a promoter that in the striatum is going to allow the selective expression of the transgene in MSNs, therefore avoiding, for instance, interneurons, astrocytes, etc. And so we could delete CB1 receptors both in direct pathway and indirect pathway MSNs. So, to make a long story short, again, we found that deletion of CB1 receptors in MSNs did not worsen the degeneration of these animals. Here simplified just by a test, a behavioral test. There is a rotarod and one of the pan markers of MSNs. So when we delete CB1 receptors overall in the mice, we have a worsening of the phenotype. But when we delete CB1 receptors specifically in MSNs, we don't see any change in the phenotype of the animals. We did, of course, the opposite approach. We did the gain of function approach. In R62 mice, as I mentioned before, in animal models of HD, the same as in patients, CB1 receptors decrease along the course of the disease. So we did just an adenoviral injection of the same CB1 receptor under the control of a neuronal promoter so that we could recover, we could rescue the levels of CB1 receptor in the striatum of these mice. You can see here, R62 mice have significantly less expression of CB1 receptors in their striatum, in their striata. And here we can rescue the expression of the receptor, but despite rescuing the receptor, we couldn't rescue the HD-like phenotype, here simplified only by rotarod. So it is curious that although CB1 receptors are massively expressed in MSNs, if we touch selectively CB1 receptors in MSNs, we don't see any change in the HD phenotypes in these animals. So we ask what may happen with corticostriatal projections. As I mentioned before, and you well know, the functioning of MSNs is critically controlled by the glutamatergic projections coming from the cortex. CB1 receptors are massively expressed in MSNs. They are very little expressed in the cortex, but we know that there is some, although very little, CB1 on cortical projections that are synaptic onto MSNs. So we did a similar approach as we mentioned before. We used this R62 CB1 receptor flux mice, and we injected an adenoviral vector with a neuronal promoter, so that is only expressed in principal neurons in the cortex, therefore about interneurons, glia, etc., encoding the recombinase so that we could delete CB1 receptors in cortical projections. So in this case, we found that upon deletion of CB1 receptors with the CRE virus in R62 mice, the phenotype of these animals was worsened. We had a worse rotarod performance compared to the CB1 expressing animals. We have less DARPP32, for instance, as a general MSN marker. And we found, and that was curious, and we can discuss this before in more detail, that this worsening of MSN viability was only evident in the D1 expressing compartment, in the direct pathway compartment, whereas indirect pathway MSNs didn't seem to be affected. So in this case, yes, when we delete CB1 receptors, not in MSNs, but in corticostriatal projections, we have a worsening of the HD-like phenotype that seems to be occurring only in the direct pathway, not in the indirect pathway. Let's go to a more subtle approach, because of course R62 mice express mutant Huntingtin in all cells of the body and all cells of the brain. So let's do just a selective expression of mutant Huntingtin in the motor cortex and see what happens when we delete CB1 receptors, specifically in glutamatergic terminals by using a LOXP-CRE approach with a next promoter so that we have CB1 receptor knockout, specifically in glutamatergic projections. So here, again, we see, that's in line with what we saw before, that when we express mutant Huntingtin, in this case solely, only in the motor cortex, we see that there is a worsening of the SD-like phenotype in the animals when CB1 receptors are lost, are deleted, specifically, again, in these corticostriatal projections. You can see here rotorod and DARPP32. And again, this is only evident in direct pathway neurons, but not in indirect pathway neurons. So again, when we delete CB1 receptors in cortical neurons, in cortical projections, we aggravate the HD-like phenotype of the animals. Is this effect of CB1 receptors located in corticostriatal projections exclusive for mutant Huntingtin, or there may be other forms or brain damage of brain overactivation in this case that can also be applied to the protection by CB1 receptors? So to make, again, a long story short, we use GQ coupled DREAD constructs to overactivate the cortex and see what happens in the striatum. So we prepare some adenoviral vectors with these DREAD constructs. We express them, these engineered GPCRs, as you well known, in the motor cortex. We injected the animals with a ligand that activates the DREAD, as you know, CNO, for instance, and therefore we can overactivate cortical projections to enhance glutamatergic excitotoxic transmission onto MSNs. So, just to give you a couple of experiments, when we treated these animals with CNO to activate corticostriatal signaling onto MSNs, we found that indeed there was a decrease in D1 and D2 neurons, but when we block cannabinoid receptors with a selective antagonist, rimonabant, we found that there was only a worsening in the loss of, in the number of D1 cells, but not of D2 cells. So again, it seems that CB1 receptors located in corticostriatal projections are the ones that are important for neuroprotection in the corticostriatal circuit, and that this occurs selectively, once again, in direct pathway MSNs, whereas the indirect pathway MSNs are not affected. So, as a brief summary of what we have been dealing with, out of the different pools of CB1 receptors located in this circuit, our data say or suggest support that there is only one of these pools that is relevant for neuroprotection. That is the pool of CB1 receptors located on corticostriatal projections that are synapting onto D1 neurons, but not onto D2 neurons. The rest of the pool, including the large pool of CB1 receptors located on MSNs, doesn't seem to be involved in neuroprotection in this circuit. Well, what happens with astrocytes? I didn't say anything because of the short time we have, but just to make a summary of what we are dealing with now with astrocytes. Of course, the endocannabinoid system is not only CB1, it comprises also CB2 receptors that we found some years ago that when they are located in microglia, that they can control MSN functionality and viability. And astrocytes are especially important for endocannabinoid degradation. So most of the activity of endocannabinoid degradation is located in astrocyte. So far what we have seen is that astrocytes in this system are controlling MSN viability not via their CB1 receptors. We have also deleted CB1 receptor in astrocytes and we don't see basically anything, but by controlling endocannabinoid availability for corticostriatal projections. That is, when endocannabinoid degradation is very low in astrocytes, we have an accumulation of endocannabinoids and we see neuroprotection by engagement of CB1 receptors located in corticostriatal projections. Whereas, on the contrary, when endocannabinoid degradation is very low, cannabinoids is very high, then endocannabinoids are low and we cannot see neuroprotection, but not via CB1 receptors located in astrocytes, but via CB1 receptors located in corticosterial projections. Yes, you will know another key neurochemical player in this synapses is the neurotrophin BDNF. And we found a couple of years ago that BDNF expression can be enhanced by CB1 receptors by a well-defined biochemical mechanisms that involve the PI3 kinase pathway. So nowadays, we are studying what can be the functional consequences of CB1 receptor modulation of BDNF expression in the plasticity and viability of corticostriatal synapses. And finally, it is very well known that CB1 receptors, and as other GPCRs, and many people here in this audience and in CIBERNED have large experience on this topic, can heteromerize with other GPCRs, can associate with other GPCRs. And for instance, in the last year, we have collaborated in the context of CIBERNED, one of the collaborative projects of CIBERNED, to see how the signaling of CB1 receptors in this system changes when it binds to adenosine A2A receptors. But we want to go a little bit further, not only study the coupling of CB1 receptors to other GPCRs, but we want to see whether CB1 receptors can couple to interactors within the cells via its C-terminus domain. And recently we have started large-scale proteomics analysis to try to find out interactors of CB1 receptors located in the cytosolic side of the receptor. And we have already a few candidates that hopefully in the next 2 or 3 years I will be able to show you about whether they can or not tune the role of CB1 receptors in these synapses. So altogether, we believe that endocannabinoids, together with other key players that are already known, such as glutamate, BDNF, etc., can be important in tuning, in controlling the plasticity and functionality of MSNs by controlling the output of glutamate and other signals from upstream, from the cortex. And just to finish, to show you our group in Madrid, the main people who have been involved in these experiments that I have been summarizing in this talk, our main collaborators, including some of the groups here in CIBERNED, for instance, Enrique Canela, Vicente Casado, a group in Barcelona, Javier Fernández Ruiz in Madrid, and the main funders. And thanks very much indeed for your attention. Thank you.
Questions from the audience? Don't be shy.
Thank you. Hi, very beautiful. So I wonder, in your experiment doing the chemogenetic stimulation, so what is the effect that you think you are inducing with the overstimulation in the motor neurons? Because I believe that you're thinking that you're having some sort of deleterious or some sort of damage. So, but I wonder, what do you know about that?
Yeah. We have used different patterns of stimulation. Of course, when you do an acute injection of CNO, you have just an overstimulation of the system in the short term. You see, for instance, an enhancement in motility, or you can see in the open field, you can see changes in general activity. And when you do an overactivation of the system, for instance, by injecting CNO for 2 or 3 weeks in the animals at high doses, you see that cortical projections are overactivated in a prolonged manner, and then you see a loss of MSNs. You see kind of 30% loss of MSNs measured by different markers, and in fact, you see that some of the functions that are controlled by MSNs, for instance, in motor behavior, are gone. We have seen, for instance, what happens when we express dread constructs in the motor cortex only in D1 neurons, based in D1 Cree mice together with flex constructs, and in D2 neurons. And essentially, we see that when we express only in D1 neurons, we see death and a loss of functionality of D1 neurons, not only on markers, but also on behavior. We see the same with D2 neurons, we see a selective loss. And when we overactivate projections from the cortex, we see a loss of both types of neurons. But when we delete CB1 receptors, we only see a worsening, a more extreme loss of markers in the D1 compartment. So we believe that from the cortex, with this system, we can tackle both subcompartments of MSNs, but when we delete CB1 receptors, we only see effects in the CB1 in the D1 compartment.
Hi, good afternoon. I have two questions. So the first one is...
Can you just introduce yourself?
Yes, sorry. I am a PhD here in Champalimod in the lab of Eduardo Moreno. And I'm not in this field of neurology, but I have two questions about this. So the first one is, do you see a neuroprotective effect of cannabinoids on Huntington disease just in preventing the onset of the disease, or they have a neuroprotective effect also after the disease is already established? And the second question is, since the onset of Huntington disease is around 40 years of age, so there is quite room of time for these people to be exposed to use of cannabinoids. There are some observational studies, or more likely case control studies about showing a correlation between cannabinoids use and the onset of Huntington disease, maybe a later onset or a milder phenotype.
Okay, thanks. There are two very interesting questions. In general, when people are outside the field, the questions are the most interesting. Yeah, regarding the first one, I would say yes. We have given cannabinoid drugs to HD mice, HD-like mice, or models, animal models of HD, and we see that yeah, there is a protection. I didn't show it, but yeah, it's not only that when you get rid of CB1 receptors, you have a worsening of the phenotype, but also when you give cannabinoid agonists to the animals, you see that there is an improvement in the symptoms and in the neuropathology, etc. And of course, the effect is better as long as you go earlier in the disease, in the course of the disease. Also because CB1 receptors, as I mentioned, are lost progressively in the disease, and it's not only known for animal models, but also for humans, not only for post-mortem studies, but also for PET studies that have been developed in the last years. So yes, the sooner the better in this case. And regarding human studies, I mean, yeah, there has been this. In fact, it's already published. There is a clinical study that was coordinated by one of the members of CIBERNED, one of the PIs of CIBERNED, that was here. I saw him before, Justo García de Llévenes, in which other groups collaborated, including ourselves, for biomarker measurements. Javier Fernández Ruiz, for instance, is another PI of CIBERNED that participated in that collaborative study. And unfortunately, we couldn't find any clinical effect regarding efficacy. I mean, the treatment was safe. We use the only medicine that is approved by the EMA, by the European Medicine Agency, so far that contains cannabinoids in its composition, that is called Sativex. That is a mixture of THC, tetrahydrocannabinol, and cannabidiol, CBD. There is another cannabinoid in a 1-to-1 ratio. And AC patients were given Sativex or placebo, and it was a crossover study. It was a short time and a small amount of patients. But unfortunately, although the drug was well tolerated, we couldn't see an effect, any symptom for efficacy. I mean, there may be several explanations for that. Of course, a mouse is not a human, and extremely more difficult to have a positive effect in a human. But there are also some issues regarding the design of the trial. We didn't have much money, we have to say, so we have to do a very simple trial, short trial, and maybe with longer times, with other doses of cannabinoids, etc., we would have had a better outcome. But so far, I have to say that the only trial that has been conducted in Huntington disease patients looking for protection was not successful. But we hope that maybe in the future we can improve the design of the trials and try again and see whether we are more lucky. Yeah, sorry. Yeah, I forgot that. Yeah. That is very complicated because, as you know, cannabis is an illegal drug, and it's very complicated to do statistics and surveys that are reliable. And Huntington's disease is, I wouldn't say a rare disease, it's a little bit over the limit, but it's also not very frequent disease. As far as we know, there are some patients that use cannabis for improvement of general quality of life. But I mean, there is no serious study that has been published on the use of cannabinoids by these patients. So maybe in the future we will have it, not so far.